Hi, my name is Kyban Savage. I'm doing A-level biology, and in this video we're going to look at how DNA codes for proteins. By the end of this we should be able to talk about how genes code for polypeptides, explain the meaning of the term genetic code, and describe the way in which a nucleotide sequence codes for the amino acid sequence in a polypeptide. To start off with, what is a gene? Well, a gene is a length of DNA, and that means that it's a sequence of nucleotide bases that codes for one or more polypeptides. In the human genome, there are about 25,000 genes, and a few of them are in the mitochondria. Now, most of these are situated on the linear chromosomes within the nucleus, and each of the genes occupies a specific place on the chromosome, and this specific place is called a locus. The DNA in the chromosome is associated with the histone proteins. Genes code for lots of polypeptides, such as the structural proteins like collagen and keratin, also hemoglobin, antigens, enzymes, there are loads. Since genes code for enzymes, they're involved in the control of all metabolic pathways and therefore in the synthesis of all non-protein molecules found in cells. So what are some key definitions that we've looked at already? What's the key definition of a gene? A gene is a length of DNA that codes for one or more polypeptides. But what's a polypeptide? Well, a polypeptide is a polymer consisting of a chain of amino acids residues joined by peptide bonds. I mentioned the word genome. That's the genome of an organism is the entire DNA sequence of that organism. The human genome consists of about 3 billion nucleotide base pairs. If you did history, GCSE, if you did like the medicine side of it, we looked at that Human Genome Project. A little side interesting note that probably made your day. Okay, so next we're going to look at the genetic code. The genetic code, well, basically the sequence of nucleotide bases on a gene provides a code, and those are the instructions for the construction of a polypeptide or protein, and the genetic code has a number of characteristics. To start off with is a triplet code, obviously triplet 3. A sequence of 3 nucleotide base codes for an amino acid, and there are 4 bases arranged in groups of 3, so the numbers of different triplet sequences is 4 to the power of 3, or 64. As there are only 20 amino acids used for protein synthesis, that's loads. It's also a degenerate code. All amino acids except methionine have more than one code. Some codes don't correspond to an amino acid but indicate stop. That's basically the end of a polypeptide chain. The genome is widespread but not universal. So for existence, the base sequence TCT codes for the amino acid serine in any organism. And this has proved useful for genetic engineering, as we can easily transfer one gene from one organism to another, and it will usually still produce the same proteins. However, there are some variations. In mammalian mitochondria, there are 2 codes for methionine, and one of the standard stop codes for tryptophan. In ciliated proctopsis, 2 of the standard stop codes code for glutamic acid, so there are variations. Well, how does a nucleotide sequence code for the amino acid sequence in a polypeptide? Genes are on chromosomes in the cell nucleus, but proteins are assembled in the cytoplasm at ribosomes. And a copy of the genetic code has been made which can pass through a pore in the nuclear envelope to the cytoplasm. A messenger RNA shortened to mRNA is this copy. Well, if we're going to look at protein synthesis, we have to start with transcription, and this is the first stage of protein synthesis. A messenger RNA molecule is made, and for this, one strand of the length of DNA is used as a template. There are 3 DNA nucleotides in the nucleoplasm and 3 RNA nucleotides in the nucleus, and then in the nucleolus. The nucleotides are activated, and they've got 2 extra phosphoryl groups attached. There are 4 different activated RNA nucleotides, such as ATP, GTP, CTP, and UTP. So if we're going to look at the different steps, first of all, a gene had to be transcribed unzips and unwinds, and to do this, the length of DNA that makes up the gene dips into the nucleolus. Hydrogen bonds between the complementary base pairs break, so it can unwind. That's it. Next, the activated RNA nucleotides bind with hydrogen bonds to the exposed bases. And these are the complementary bases. For example, U binds with A, G with C, A with T. As usual, we learnt last year on the template strand. And this is catalysed by the enzyme RNA polymerase. The two extra phosphoryl groups, phosphates, are released, and this releases energy for the bonding adjacent nucleotides. The mRNA produced is complementary to the nucleotide base sequence on the template strand of DNA. Therefore, is a copy of the base sequence on the coding strand of the length of DNA. Finally, the mRNA is released from the DNA and passes out of the nucleus through a port in the nuclear envelope to a ribosome. Now, that was a lot of information and a lot of words. Basically, it's just what we learnt last year in AS level biology, but just with a whole lot of more words thrown in. So, yeah. Thanks for that. I'm retaking the summer exam. I can't remember which one that is, F2, blah, blah. I'm retaking it, so I'm going to AS lessons again, which is really helpful, actually, because I've completely forgotten a lot about DNA. So when I'm back in the lesson, I'm just like, oh yeah, that does happen. Oh yeah, I should know that. So now, yeah, it basically does help for this exam. So if you get out your AS level textbook and look through it, it'll be good. If you want some questions, look in your book, or I'll read them out or give you some. You could explain why the genetic code would not work if nucleotide bases were arranged in pairs rather than triplets. Also, you could say what is meant by a degenerate code, because I said that earlier and didn't give a definition. You could also define transcription. Yeah. Yay! So that is the end of this video, and I hope you know all that, and good luck with your biology exam.